Hey guys, we're picking up here on page 9 of the notes. We are going to have an assignment today. There's not that many pages left of the notes that I want to do today, but I'm going to kind of walk through how to do the assignment. So that's where we're at, I guess. The codons, remember, are the 3-letter sequences on the mRNA. Remember, that's the message RNA. And the anticodons are on the tRNA. And remember, they have to kind of fit together. Okay, so I'm just going to kind of flash back to the last page in case we kind of forgot, because this was a while ago. So here's the mRNA. This was a copy of the instructions in the DNA. Each one of these little light blue things is a tRNA. There's an anticodon. There's another anticodon. And so remember, each tRNA brings a particular amino acid. And we are putting the amino acid together in a long chain to build a polypeptide, which could become a protein. Okay, remember, polypeptide's like a baby, immature protein. But the key idea here, the unicodons and anticodons are the same for all organisms we have ever examined. And I just want to kind of point out to you how crazy that is, right? So there's millions of different species, and in every single species, if I have the same codon, I am going to get the same anticodon, and from that anticodon on the tRNA, I'm going to get the same amino acids. Okay, from everything, from bacteria to plants and animals, everything. This has allowed scientists to use bacteria to produce useful things for humans, like insulin. Okay, so we take a gene from humans, which is instructions for the protein of insulin, and we take that gene and we put it into another organism like a bacteria, and the bacteria can read those instructions and make insulin, no problem, right? Because their machinery to read instructions is the same as ours. Okay, we can also use this to modify crops to be herbicide resistant, and specifically the one you probably heard of here is Roundup. If you ever hear something, a certain seed variety called Roundup Ready, that means they have inserted a gene from some other organism so that it can now make a protein that breaks down this herbicide. And so it can survive when sprayed by it, and all the weeds die, but it gets to live. And so for this reason, it's called the universal code. Okay, since it applies to everything. Universal means it can be used all over the universe. So it's a universal code. And so we have this idea called the central dogma. Okay, and if you have like a personal dogma, it's like something that you believe to your core. Like somebody's like, oh, my central dogma is, be kind to all, you don't know what they're going through, or something that you like centrally believe. And if that if that was untrue, your entire world would fall apart. Okay, well, for biology, our central dogma, this kind of idea that this is how biology works, is this. Okay, we take DNA. From the DNA, we make RNA. And then the RNA is able to make the protein. This 3-part little chain here is how all living things basically function. Okay, so this first part from DNA to RNA, remember that step is called transcription. Okay, the DNA is transcribed or just written down as RNA. And then from RNA to protein was translation. Remember, the easy way to remember which one's first. C comes before L. They both start with trans. Right, and then we have C comes before L, transcription before translation. And then I have one thing I always kind of like to add on here is a loop from DNA to DNA, and that was called replication. Okay, so these 3 things help organisms do everything. Okay, so DNA's instructions for building protein. But between the instructions and the final product, we need somebody to do all the work, right? And then replication is we got to make new copies of DNA for each cell. So those are our three very, very important processes in this chapter. On the back, we have this big, beautiful table, okay, which is the universal code. Okay, so I said, remember, the code is the same. Well, what's the code? Here it is. Okay, and these are codons, which, remember, are on the mRNA, the messenger. Okay, so we're going to practice with this a little bit on the next page, but just kind of one time through here. On this kind of a diagram, we read from the inside out. Okay, so you don't have to write this down. We're going to have some practice it with a little bit. But let's say my codon said UCA. And I want to know what amino acid does that codon call for. Reading from the inside out, I find the U, and then the C, and then the A, and this is the amino acid. Okay, so we would want to stay maybe star here. Outside words are amino acids. Okay, there's 20 different ones. And if you notice, some of them would have more than one codon. For example, the one we just looked at, serine. I would have UCU is serine, UCC also serine, UCA also serine, UCG also serine. Okay, so that is true. So let's kind of do a little practice here. All right. So it says, let's say a sequence of DNA reads blah, 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 blah. All right. What would be the mRNA sequence transcribed from this? And we're going to ignore introns. Remember, introns are the kind of junk DNA that after you transcribe, you would cut out. Okay, but we're going to ignore that for right now. So we're going to transcribe this, which is DNA, to mRNA. So T, apples in the tree, so that'd be A. G would go to a C. C to G. And again, I would encourage you to pause this and try it, and then check your answers when we're done. But that's up to you. I'm just going to kind of put a little thing under here to make sure I don't get lost. A, so A usually would go with T if I was making the other side of DNA, but since we're doing RNA, I put in a U. UA. Okay, hopefully I didn't make any mistakes here. So this would be my mRNA. So if this is what my mRNA would be, come out of the nucleus and find a ribosome so we can get down to business here. And it says, what would the tRNA be? Okay, so tRNA, remember, is these guys. So we're saying what sequences on the tRNA would match up to the RNA. It says don't start until you get to a start codon for methionine. Okay, and I don't, that was the other day. So remember, methionine is always where we start, always. Okay, that's always the amino acid that is first. So I look back at my chart here, and I find methionine. Let's maybe label that start. Okay, so methionine is AUG. Remember, always read from the inside out. So that is where we want to start. So I'm kind of scanning here. Here's an AUG. So that's actually where I would start. I wouldn't start translation. Remember, that's where the tRNA brings the proteins or the amino acids in until I get to a start. So I'm not going to worry about these three. I'm going to start right here because that's for methionine. That's where we start. and here we go. So then this is complementary. So we're RNA still, so U's instead of T's. So A with U, A, C. Again, I would encourage you to pause, try this on your own, and then double check against me. But here we go. Alright, so there is my tRNA. Now, so this was that first process. This would be transcription. And then translation is actually putting amino acids in a chain. One kind of cheat that I will allow you, because we did this in my college classes, so I don't know why we couldn't do it now. Instead of writing out the full name of the amino acids all the time, if we just write the first 3 letters, that's going to be okay. But then if it's an acid, okay, make sure you put acid, because aspartic acid and asparagine both start with ASP. Okay, so that's the only one I think you're going to have to be really careful of with the first 3-letter idea, is that aspartic acid. So, these are, remember, the codons, which are the mRNA. So I'm going to start here. Remember, this is, I said we're going to start here. So AUG, I already know the answer to this one, but that's okay. AUG is methionine. So I'm going to write met. That's my first amino acid. Second, I go to the next 3, A, G, C. And I'm going to kind of split these up. So again, anything so you don't kind of get confused or lost here. So A, G, C. Reading from the inside out, A, G, C takes me to serine. There's my second amino acid. Okay, third amino acid, UAG. Start from the inside out. UAG. That's a stop codon. So that's actually it. That's the end. I probably didn't do that on purpose. Okay, so this would be my entire polypeptide, 2 proteins. So when you get to stop, you stop. You're not like, well, I'm just going to keep going. No, stop means stop, right? So notes-wise, that's where I want to stop. I'm going to look a little bit at your assignment here. This is on Google Classroom. I would really, really strongly encourage you to read this through, but I'm just going to kind of make this, try not to make this video too long. So, how to decode messages. So basically, we're going to pretend the DNA, instead of being that the message in the DNA is, you know, for a protein, we're going to pretend the message in the DNA is like a message, like one that one might send another person. Okay, so if this is the DNA, we're trying to figure out the message that the DNA is. Okay, so to do that, we made each amino acid represented by a different letter. All right, so let's just give it a look here. So their example starts like this. So this was the DNA. All right, so this is their example. And so they transcribed it, right? So they made the mRNA. That would come from the DNA. And it starts with the AUG, right? I don't think they will all necessarily start with the AUG for the purpose of this code breaking activity. Just so you know. So then they went, okay, so AUG is for methionine. There we go. GCA. GCA is for alanine. And they just kind of went through till they got to a stop. Okay. Then they will use this code breaker to turn the amino acids into a message. So methionine, if I'm methionine, is an M. Okay. So I would go M. Alanine, look at my code. Alanine is an A. Lysine, look back at my code. Lysine is a K. And glutamic acid, my glutamic acid is an E. Okay, so the first word of this code is make. Alright, the first word of that code is make, which is what they got. If I can force the right page here. Oh, there it is. Okay, the first word's already done. Make. So we could finish this. Let's just finish this one. Okay. So and I would encourage you find some way to keep your 3 letters straight from each other. So TAC would be AUG. ATA would be UAU. ACT would be UGA. I'm just going to do this. CTG would be GAC. CGA would be GCU. And ATG would be UAC. Alright, so let's see here what those would come out to. So AUG is methionine again. UAU, try to make this all fit on here. Yes. UAU is tyrosine. And UGA is stop. Okay, every time you get to a stop, it's going to be a space. Okay, so we have make, and then met, methionine and tyrosine would be M for methionine, and tyrosine Y. So that'd be my stop space. Okay, so that's methionine, tyrosine, stop. And then we have GAC. Skipping ahead here. GAC, aspartic acid. Okay, GCU, alanine, and UAC, tyrosine. Alright, so we have 3 left here. Aspartic acid is a D. Alanine is an A. And tyrosine is a Y. Make my day. Okay, so that's what this code was trying to say. Make my day. So I really like this activity because if you get done with it and you get a code that doesn't make any sense, that's a really good clue that you need to go back and try it again. I do think on yours, I cut this last one off of there. If you would like to write on a separate sheet of paper, if you're like, this is not enough, or you hate the typing part, feel free to write this on a separate sheet of paper and take a picture of that and upload it. But so your assignment is these 4 that are on the Google Doc, and there are 3 kind of thinking questions. And so again, if you want to do this on a separate sheet of paper, go for it. Otherwise, feel free to type it out. It might be easier to do it on another sheet of paper and take a picture, but I'm going to leave that up to you. Good luck. This one is usually kind of tough, and I usually have this be something that people work on in a group of 2 or 3. So I will expect questions from you. Please, 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 please ask them. Don't just guess. Don't just guess. Ask your questions. Get them answered. And good luck to you. Talk to you later.